morning. Thank you so much, Adeline, for inviting us here and giving the opportunity of work with such competent speakers that we'll introduce soon. I'm Gustavo Maegawa. I'm in Columbia University. I work with patients as well and research. We have a research lab working in small molecules for Krabbe. And we're going to have a session here with one of the representatives of the advocacy groups, Lesa Brackbill, and from Hunter's Hope. And then Dr. Shin is going to, from the University of Buffalo, is going to talk about the pathogenesis in Krabbe. And then finally, Dr. Bradbury was going to talk to us about the significant advances in preclinical models as well as gene therapy. Here is our work group. There were several meetings, at least 3 meetings that we discussed the content we're going to present today. So Krabbe disease is a disorder caused by the deficiency of galactosyl-ceramidase or galactosyl-cerebrosidase. It's caused by biallelic variants or pathogenic variants in the GALC gene. There was significant contributions by Dr. Wenger, David Wenger, who is here, to the understanding and cloning of the gene, the identification of the first pathogenic variants, and also the patients. So this disease is very old, is over 100 years old, described, and significant advances have come through today. So the most prevalent form is the infantile form, which presents between 0 and 6 months of age. A rapidly progression of the demyelination and significant compromise in patients that ended up dying between 1 and 2 years old. The late onset forms are now recognized today, and more patients are being diagnosed and identified with those forms. So from the pathology standpoint, the classical form or classical pathological hallmarks is those multinucleated cells, the globoid cells, therefore globoid cell leukodystrophy, and a diffuse demyelination, frontoparietal and occipital demyelination occurs in secondary atrophy, diffuse brain atrophy. And the culprit is the substrates of GALC. One of them is cycosine or galactosyl-sphingosine, which has a significant reactivity to this free amine here located in the carbon 2. Significant cytotoxicity for those cells producing myelin, the CNS oligodendrocytes and Schwann cells in the periphery. In terms of the research, the challenges and successes are stated here. We discussed the significant advance in terms of the research basic, the translational as well as clinical. There was significant development of animal models, the murine, canine, lately non-human primates that led to the understanding of the late onset form of Krabbe disease. Multiple modalities of therapy are now being investigated, targeting different pathogenic mechanisms of the disease. The challenges still today remains. So there is secondary pathogenic cascades that still we don't understand which ones prevail in different forms of the disease and which needs to be targeted when developing therapies. Limitations of preclinical models, some of those are more representing the infantile, the early onset. Very few models that we can rely on developing late onset therapies. So the understanding of the natural history is still lacking, so we still need to know the natural history of treated patients undergoing bone marrow transplant, as well as those with late onset forms. There is also cycosine has been very helpful in the newborn screening, but very difficult to monitor patients after bone marrow transplant or treatment. In terms of clinical grounds, we have a very strong advocacy group that has been resulted in newborn screening in multiple states across the nation. We have very strong centers of excellence coordinating diagnosis management. But still challenges. We need to expand the centers of excellence, educate ourselves, physicians, healthcare providers, and the community about Krabbe. There is a lack of guidelines depending on the form of Krabbe. We have some attempts for especially patients being transplanted, but not standards for late onset patients, for instance. Biomarkers are still under discovery, especially when therapies are coming along to clinic gene therapy. And there is a lack of understanding of who is going to have access to therapies across the worldwide. Patients being diagnosed that maybe some of them will be benefit from therapies, others may not, and we don't know specifically how many patients are outside the U.S., and just as an example. So those are the heat maps. It's still improving over the years since 2019. The hallmarks here that still need work: therapeutic development, advocacy group in terms of looking at states that are still on the not having the newborn screening, therapeutic development for patients who have late onset juvenile forms. In terms of the management of patients, access to disease-modifying therapies are important. More expanded access to clinical studies as well as to the mainstay therapy, which is the bone marrow transplant for early infantile. And also a better understanding for us in terms of the disease, the guidelines for patients who have late onset juvenile forms of the disease. So here I'm going to pass along to Lesa, who's going to talk to us about the advocacy groups. Lesa, please.
Good morning. My name is Lesa Brackbill, and I am the Outreach Coordinator for Policy and Advocacy for the Leukodystrophy Newborn Screening Action Network. Come see me for a button later. Most importantly, I am the mother of a child who had Krabbe disease, and I'm also the parent advocate behind Act 133 of 2020 here in Pennsylvania, legislation that reformed the newborn screening program and led to the addition of Krabbe to the state's newborn screening panel. Since then, we've identified 4 babies with Krabbe disease. I was honored to be asked to present on the landscape of Krabbe-specific advocacy organizations and have been given information from each respective group to share. These are only highlights, of course. Each organization does more than I have time to share here. We are fortunate in the United States to have many advocacy groups that focus on Krabbe disease, but that is not the case in much of the world, and this is certainly a need. Today, I will present information from each group in alphabetical order. Each organization is filling different needs of the scientific and patient community. However, we've also been working together on common goals when possible. Before I share about each organization, I wanted to provide an update on newborn screening for Krabbe disease. This data is from July of 2022 and came directly from the state newborn screening programs. As you can see, only 10 states are currently screening. We are hopeful that Krabbe will be added to the Federal Recommended Uniform Screening Panel, or the RUSP, in 2023, and when that happens, the states in yellow will add it to their panels within 2 to 3 years. The states in orange have laws in place to screen for Krabbe, but have yet to implement. Before I speak about each organization, I wanted to briefly mention the Krabbe Stakeholders Group. This was formed in 2021 in an effort to encourage collaboration between advocacy, industry, clinicians, and researchers working in the Krabbe disease space. We are stronger when we work together and share resources, and that is what we are striving to do with this initiative. Hunter's Hope was started in 1997 by Jim and Jill Kelly in honor of their son, Hunter. Since then, they have worked to support families, advance newborn screening, and improve care for patients with leukodystrophy. One of their primary areas of focus has been changing how patients with Krabbe disease receive care by creating a network of centers known as the Leukodystrophy Care Network, or LCN, which was established in 2015. Each center must meet specific criteria. Among other things, this includes a multidisciplinary team to provide comprehensive and proactive medical care. There are currently 12 LCNs and 4 affiliate centers. Hunter's Hope's efforts for expanded newborn screening for Krabbe and other leukodystrophies has several components with the ultimate goal of nationwide and eventually worldwide newborn screening for Krabbe disease. They are involved in both federal and state advocacy. In July of 2021, with the help of the RUSP Workgroup, Hunter's Hope resubmitted the nomination package for Krabbe's inclusion on the RUSP. In May of 2022, the ACHD&C voted unanimously to move Krabbe to full evidence review, and that review is currently underway. The committee will vote in February of 2023 regarding Krabbe's inclusion on the RUSP. And finally, Hunter's Hope has worked for many years to publish clinical practice guidelines to ensure that accurate information is available to every provider, regardless of their expertise with leukodystrophy. To date, 4 CPGs have been published. Krabbe Connect was started in 2017 to bridge the gap between science and patient knowledge. Their vision is a day in which each patient receives an early diagnosis, has access to state-of-the-art care, and lives a life free of Krabbe disease. One of the ways Krabbe Connect serves families is through their patient assistance program, which has several components. This includes a holiday grant program to help families make memories with their affected loved one. They also have a patient support team for newly diagnosed families. Their Partner With Us program gives families the opportunity to establish a family-advised fund utilizing Krabbe Connect's nonprofit platform and resources to strengthen groundbreaking research, contribute toward a special project, or simply to aid other impactful efforts in the Krabbe disease space. There are currently 4 family-advised funds. Krabbe Connect worked together with NORD to implement the Krabbe Cures platform. Krabbe Cures is a community-owned engagement study that will provide patient-reported information to further understand the burden of Krabbe disease and to obtain IRB-approved data for patients and caregiver perspectives on transplant and gene therapy. This study can be amended to add additional surveys as the engagement study progresses over time. They are also working with us at LDNBS to create fact sheets profiling Krabbe families in all 50 states. 8 were created last month. The Leukodystrophy Newborn Screening Action Network, or LDNBS, was started in April 2020 and has 3 main objectives: advancing newborn screening for leukodystrophies and lysosomal storage disorders, supporting newly diagnosed families, and encouraging collaboration. We seek to equip advocates who want to see their condition added to their state's newborn screening panel and offer numerous tools and resources to help them succeed. Our current focus is on the 3 leukodystrophies with newborn screening tests, which are XALD, Krabbe, and MLD. One of the tools we offer are up-to-date maps for those conditions to show the status of newborn screening for each. We also have a page with international information, but there are no other countries currently screening for Krabbe and only pilots for MLD at this time. In addition, if you click on an individual state, you can learn more about their particular programs and other useful information to help you advocate. One of my favorite resources we offer is a detailed spreadsheet with data for each state that can help advocates better understand how their state operates. Each column has a note explaining why this information matters and how it can help them. While this particular project is not Krabbe specific, it did lead to a partnership with Krabbe Connect to make fact sheets profiling Krabbe families. These fact sheets are for the families and advocates to use as they make connections with stakeholders, and we share them as well. And finally, we help advocates discover how to best advocate for newborn screening in their state by using these 3 steps detailed out on our website. Each of these areas are important to fully understand before beginning advocacy efforts. As an example of how we equip advocates, later this month we are hosting a Facebook Live with some newborn screening advisory board members from 4 different states to help reduce intimidation and help advocates feel more comfortable approaching the board in their state. Partners for Krabbe Research was founded in 2012, and their mission is to increase awareness and support research to improve the lives of those born with Krabbe disease. Their primary focus is to fund resources for the NDRD Brain and Tissue Bank at the University of Pittsburgh. They cover the expenses for donation and guide families through the process. Having been through this process personally, I can attest to the care taken every step of the way. The NDRD Brain and Tissue Bank is run under the direction of Dr. Julia Koffler at the University of Pittsburgh, and it receives donations from affected families, and those tissues can be used by researchers. To date, 21 donations have been made to the biorepository. Brain donation is of critical importance in furthering our understanding of neurodegenerative disorders and provides a gift of hope to future generations in the fight against Krabbe and other similar diseases. This brochure for families who wish to donate to the Brain and Tissue Bank can be found at the website below. There have so far been 3 manuscripts published using the brain and tissue samples from the biorepository. 2 additional papers are in the works and will be published in early 2023. These generous donations from families are driving research forward and lead to new discoveries about Krabbe disease. And finally, the Legacy of Angels Foundation was started in 2008 by Paul and Sue Rosenau. Their mission is to improve the lives of children by working to promote the expansion of newborn screening and to further education, awareness, and research of Krabbe disease and cystic fibrosis to provide a better treatment and a cure. The Legacy of Angels Foundation is dedicated to filling gaps in Krabbe disease research by providing grants to scientists, and since 2009, they have funded around $20 million in grants. In addition, the Legacy of Angels Foundation hosts the Krabbe Translational Research Network meeting and has held 10 annual meetings to date. This meeting is primarily attended by scientists and researchers, and they present their most recent findings and publications. The future is collaborative, and the Krabbe disease patient advocacy groups and nonprofits are working toward that goal. Resources are limited and time is of the essence, but by working together, we can make a difference and achieve our common goals. Thank you.
Good morning. My name is Daesung Shin, SUNY Buffalo. I'm going to talk about the pathogenic mechanisms of Krabbe disease. So I'm going to talk about what is known and what is not known in Krabbe disease study. The Krabbe disease is autosomal recessive lysosomal storage disorder involving both CNS and PNS demyelination. 1916, Dr. Krabbe first described the disease with the multinucleated cells. That is the pathological hallmark of Krabbe disease. In the US, the incidence estimated about 1 out of 100 to 250,000 people a long time ago, but recently updated incidence in New York State newborn screening said in case of the infantile Krabbe disease, it is about 1 in 394,000 babies, but it could be much higher, including if we include the later onset forms. There are two forms of the disease, infantile onset form and later onset form. Infantile onset form is normal at birth, but symptoms begin before age 1 with this phenotype, and mostly fatal before age 2 to 3. Later onset form shows slower disease progression with this variable clinical course from 1 year to the 7th decade of their life. Weakness, muscle stiffness, and difficulty of working, vision loss, intellectual regression, and dementia are showing. This is a 9-month-old infantile form Krabbe baby. You can see some spasm of muscle and then clenching of both hands. And this is the 7-month-old MRI with the control. You can see some of the volume loss and typical abnormalities in the thalami. The global cell is the prominent feature in the Krabbe patient. This is reviewed nicely 2021. This is in both CNS and PNS. You can see in the red circled area, you can see the multinucleated big large cell. We call it this is global cells. A similar cell type also can be found in the PNS system. This disease is caused by a mutation in the galactosylceramidase GLC gene mutations, and this is some of the mutation list found mostly all the 17 exons. You can see most of the mutation or the cis polymorphisms can be found like this way. Normally, the GLC catabolize galactosylceramide and galactosylsphingosine, if you see this one, to generate galactose with sphingosine and ceramide. Galactosylceramide is a major lipid in the myelin sheath. If you see this figure and these high-powered images, this outside the lipid membrane is mostly GLC. And the galactosylsphingosine, also called as the cytosine, that may cause the cell disease and cell death and disrupting the lipid raft and causing oligodendrocyte death. So if you see this, in case of the Krabbe disease patient, due to the GLC protein deficiency, the cytosine cause oligodendrocyte death and demyelination. This demyelination generates more the galactosylceramide. It can elicit more macrophage reaction. This is currently known etiology of the Krabbe disease. The GLC precursor protein is generated in ER with 80 kilodalton and processed in the lysosome. Some minority is directly secreted to the extracellular space and then uptaken by the same cell or neighboring cells. The maximal enzyme activity shown around the pH 4.5, saying that GLC is a lysosomal enzyme. Recently defined the GLC protein structure said there are 3 large big domains. And so the mutations found everywhere means that depending on the location of the mutation, it can affect the GLC proteins like processing or the enzyme activity or the targeting to the specific subcellular organelles. So I said that some of the proteins is directly secreted outside of the cells, and then the uptaken by the secreted cell by itself, or the uptaken by neighboring cells. This is known to be a mechanism of the currently available therapeutics like transplantation, ERT, or gene therapy. So for the uptake of this uptake process, they need some of the membrane receptors named mannose receptor or mannose 6-phosphate receptors, which means that those 2 proteins targeting on the plasma membrane might be key to make more efficient uptake. Another recent paper published in 2015, they tested uptake efficiency with this ERT in newly generated Krabbe mouse model. The red color is GLC protein and the green color is cell-specific markers, the macrophage, astrocyte, neuron, and oligodendrocyte. If 2 color is overlap, means that more GLC protein is present in specific cell type. If you see that the red color is all in boundary of the green color, macrophage, astrocyte, and even neurons. But in case of oligodendrocyte, you cannot see much overlap both colors, means that there might be differential efficiency of the GLC uptake. This slide looks complex, but this is about the cross-correction is impaired in the GLC mutant. This is Western blot, the purified Schwann cells. Schwann cell is the peripheral myelinating cells. So we can purify those specific cells from the Krabbe disease models and then mix it with exogenously expressed proteins in the culture medium and then see how much those proteins can be uptaken inside of the cell. So if this band means that this how much is the GLC proteins inside of the cell. The plus, minus means the genotype. So you see that the wild type knockout, wild type knockout, wild type knockout. You see that the wild type is higher than the knockout. And this is quantification, means that in case of the mutant Schwann cell, they have less cross-correction efficiency. Similar experiment is done using the transwell system. This is the co-culture system. So you can culture 2 different cell types and separate place at the same time. And the cells was purified mouse embryonic fibroblast from the twitcher that is the well-known the Krabbe disease model. If you see compare these 2 bands, the wild type has more uptake in the GLC, but in case of twitcher, it's much less. Same experiment is done the human fibroblast with the normal control, late onset Krabbe patient fibroblast, infantile onset Krabbe patient fibroblast. Also, you can see that the band intensity is much lower in case of the Krabbe disease patient compared to normal control. This slide is showing that the cross-correction might be impaired in GLC mutant. So the 2 galactolipid named psychoshin and galactosylceramide is highly increased in the Krabbe patient brain and mouse model. Especially the psychoshin toxicity is extensively studied in the Krabbe scientist community. Especially in this model, for example, how the psychoshin toxicity can be generated. There's a name that lipid RAP2 is a specific protein structure domains present in the plasma membrane. They are quite important for cell signaling and cell homeostasis. But in case of the Krabbe patient, the psychoshin is highly accumulated in this domain and disrupt this structure, and therefore the signal activity can be downregulated and some myelin stability is down. Eventually, cell can die and neurodegeneration can happen. This is one of the models how the psychoshin toxicity can be generated. Besides this, there are many other cytokine toxicity studied. I'm not going to explain all of this, but what I want to say is that scientists in our community seriously have studied about this cytokine toxicity. But it looks like cytokine may not be sufficient to produce all of the things of the Krabbe pathology, at least in the PNS, peripheral nervous system. So by GLIA system, we can specifically delete the calcium proteins generation in the Schwann cell specifically. This is the peripheral nervous system myelinating cells. And this mouse model has a good phenotype. It has the peripheral neuropathy as shown in the Krabbe patient. And however, the phenotype was much milder. There was no survival change. And, they live like the wild type. And this is one of the example that macrophages phenotype. This is the Krabbe full lockout mouse model macrophage. You can see the very large macrophage having lots of debris. This is a wild type control. You can see the normal shape of the macrophage. But the Schwann cell conditional lockout mouse model looks like the wild type, not much looks like a knockout. However, the cytokine level when they measured in the sciatic nerve, the quite similar level you can see like this, which means the cytokine, besides the cytokine, there must be something else to produce this Krabbe pathology in the PNS. Until recently, it was not much known how much the calcium protein is expressed in each different brain cell type. Recent study showed that this red color is a cell-specific marker and calcium protein marker. This calcium protein is expressed in all different cell types. This is neuron, oligodendrocyte, IBA1 means that microglia and the astrocyte. And this is quantification, especially very young mouse brain. The most of the calcium protein is expressed in neurons, which means, but so far, because of this extensive demyelination in this Krabbe patient, we thought that the oligodendrocyte is the primary cell type affecting this disease phenotype, but it is possible other cell type also can independently contribute to the disease. For example, one of the recent studies showed that neuronal calcium ablation using the same GLIA system, and this mouse nicely shows some phenotype, body weight loss as time goes on, and some of the neurodegeneration phenotype. You can see lots of vacuoles in the mutant, and some of the debris accumulated in the axons. And the neuronal cell number is much downregulated like this, normal control, and this is much less. And neuroinflammation also increases like this way, suggesting that neuronal calcium ablation also has its own cell autonomous function of the calcium. So the deletion of the neuronal calcium can elicit the neurodegeneration. And calcium deficient neurons can produce cytokine. Two independent groups tested this cytokine generation, calcium neuron-specific calcium knockout mouse model, and purified the neurons. Neuronal-specific the calcium deletion mouse model, if you see this green, this means the cytokine level up compared to the control, although its level is much lower than full lockout mouse model. In the purified neurons, you see that oligodendrocyte, of course, it produces lots of the cytokine. However, the neurons also can produce cytokine like this. Calcium is another substrate of the calcium protein, and this is done in the peripheral nervous system macrophages. So we can purify the macrophages from the bone marrow of the Krabbe mouse model and treat the calcium and to see how the knockout macrophages can respond to this provided the calcium. And this is wild type control, and this is knockout macrophages. The green signal means that the calcium is accumulated inside of the macrophage. And if you compare to this cell shape, you see the much globular shape and much larger, means that this is globose cells. If you measure some of the inflammatory cytokine level, for example, the TNF-alpha and IL-1 beta is quite representative of the markers. You see that only the calcium treatment increased this level, but cytokine is not. Besides this, the globose cells has some effect in the myelin degradation, although I didn't provide any here. So, Krabbe disease may be caused by at least two mechanisms in the peripheral nervous system. First, as we know, cytokine-induced demyelination. Secondly, a secondary neuroinflammation from the GAL cell storage in macrophages. This is a neuroinflammatory process during the Krabbe disease lifespan and some other animal models as well as in the Krabbe patient. This is lots of things done to study about neuroinflammation increase in each the ages, and especially the microglia and the macrophage and astrocyte are critical to generate this type of the neuroinflammation. And this is the final summary figure that how the disease can be caused. By the calcium protein deficiency, cytokine is generated from the calcium by the isolation in the oligodendrocyte, and increased cytokine can cause the neurodegeneration. The degenerated myelin debris and the neurodegenerative product can be phagocytized by the macrophages or the microglia, but the macrophage or microglia cannot process this eaten product because it has also no the calcium proteins. So it can be more pro-inflammatory, and the inflammatory cytokines can be secreted like this way. Neuron has its own problem besides this the secondary effect, because the neuronal lysosome is quite important for the axonal transport and cytoskeleton maintenance. So due to this calcium protein deficiency, it has its own problem. So this is a summary. The calcium mutation in Krabbe disease accumulate GALCA and cytokine, and cytokine is generated from GALCA by the isolation, and cytokine accumulation in membrane triggers, of course, cell death. And GALCA accumulation independently can result in global cells and inflammatory reaction. And both the calcium deficient oligodendrocyte and neurons produce cytokine. We still are studying about more specific function of the microglia, macrophage, and astrocyte. And cross-correction is not much efficient in calcium mutant cells. This is, I like to appreciate all the Krabbe working group, people who helped this study, and the collaborator in the University at Buffalo, and some other collaborators, and then this is my funding source. Thank you.
Hi, I'm Allison Bradbury. I'm a principal investigator at Nationwide Children's Hospital and assistant professor in the Department of Pediatrics at Ohio State University. It's really great to be back in Philadelphia and share with you guys today some of the preclinical and clinical development in Krabbe disease. I really wanted to look back at our heat map and just try to focus on two key successes and two key challenges in Krabbe disease. So looking at our highest score in our heat map overall, scored 4.5 in the availability and utility of preclinical models. And secondly, looking at the therapeutic development, the second highest score in one of our heat maps is the active clinical trials. So as was just nicely presented, there's a really long history of Krabbe disease, which was discovered in 1916. There's been discovery of numerous animal models, including a canine model of Krabbe disease, a murine model of Krabbe disease, and a primate model of Krabbe disease, with numerous key preclinical studies happening along the way. Many of them initially looking at bone marrow transplantation, substrate reduction therapy, and then really over the last 20 years, it's been a theme of combination therapies, looking at gene therapies combined with bone marrow transplant or substrate reduction, or really all of them in combination, as we've learned more about really how challenging it is going to be to tackle Krabbe disease. So what are the preclinical models of Krabbe disease that are available, and why was this our highest score in our heat map? So first off, we have a number of patient-derived in vitro models of Krabbe disease. Let me go back to that. If we just look at the Coriell models, there are over 30 models. Sorry, this seems to be a little glitchy. Over 30 different lines of Krabbe disease. These include iPSCs, fibroblasts, and a number of lines involved in biobanks from NIH and others. We also have a zebrafish model, and this was developed by Marco Presta's group in 2014. There's 2 genes in the zebrafish, GALC-A and GALC-B, so they used the Morpholino technology available at the time and knocked out those genes and crossed them. This was kind of a more mild model that had no cytosine accumulation. And so right now, Laura Feltry, who is one of our Krabbe working group members, shared that they're working on using CRISPR gene editing and making a pure knockout of these 2 genes and crossing those fish to really study more about mechanisms by the zebrafish, and this is funded by the Legacy of Angels Foundation. So we also have a number of spontaneous models of disease. So we're really fortunate in this regard that the first is the Twitcher mouse model, and this has been extensively studied and used in preclinical studies. We also have a feline Krabbe disease that was published but has not been really well characterized, and a colony was not maintained. In contrast, we have a canine model of Krabbe disease. That's a missense mutation that is also naturally occurring, and a colony of the dogs was maintained here at UPenn after being identified by David Wenger here in the audience. There's also been a sheep model of Krabbe disease that was identified and published, but again, not fully characterized and not maintained. And then there's also a non-human primate model of Krabbe disease. Although this most closely recapitulates human biology and size, this really isn't a tractable model due to the fact that it takes 3 to 4 years to reach sexual maturity. They have 1 litter a year of usually 1 animal, and with this being an autosomal recessive loss of function disease, you need statistically a quarter of your animals. So it would take really an enormous undertaking to have a colony of non-human primates, although a few of these have been studied in some preclinical models. In addition to our spontaneous models, there have been a number of genetically modified mouse models made over the last 20 years. Some of these are representing later patient onset, some more juvenile onset mutations. We've also had some really interesting technology with trying to lysosomal membrane tether GALC or express GALC in just specific cell types. So these are newer models that are also really important and critical in understanding some of the mechanisms of Krabbe disease. So really, I think one of the highlights of this being in our heat map is that we have these patient-derived models. We have zebrafish, which are great for screening. We have the naturally occurring Twitcher mouse and the genetically modified mouse models. And we also have this large animal model that really allows us to study some of the more clinically relevant readout measures and ways to deliver therapy. Krabbe also has this advantage that for many years, the standard of care has been hematopoietic stem cell transplantation. What we've seen over the years from the transplantation is that asymptomatic patients really benefit in survival and cognitive neurodevelopmental outcomes. The classic infantile babies that are diagnosed prior to 12 months of age, if they are transplanted after the onset of symptom, there really is little to no benefit from the transplantation. So it's really no longer indicated for the infantile patients. So the goal now is to diagnose and transplant the infantile cases by 4 weeks of age. Some of the later onset babies that are diagnosed later and have symptom onset after 12 months, they do have some benefit from transplantation still. But one thing that's been shown over the years, even when we follow the asymptomatic transplanted babies long term, they still end up needing walking assisted devices or wheelchair bound due to the fact that transplantation does not completely correct the gross and fine motor skills, and it does not efficiently target the peripheral nervous system. So a lot of effort over the years has been how to improve upon the standard of care. So one clinical study that's been going and Joanne Kurtzberg group at Duke University is this DUOC study where in addition to the umbilical cord transplantation, a second lot of cells is derived into these oligodendrocyte-like cells and delivered directly into the central nervous system. And this is supposed to help increase how rapidly the cells engraft to enhance remyelination and also just more direct targeting of the central nervous system. So that's ongoing at Duke University for a number of years. But really, one of the things I wanted to highlight is that this year has been huge for Krabbe, and that there's been the initiation of 2 gene therapy clinical trials after work that's been ongoing for a number of years. So I just wanted to briefly highlight the major differences in these trials because, people are like, wow, 2 trials in 1 year. They are very different. So one trial is using the standard of care, hematopoietic stem cell transplantation, with preconditioning and unrelated umbilical cord blood transplant, followed by an intravenous or systemic delivery of AAV gene therapy, and the serotype is AAV-RH10, and this study was licensed to Forge Biologics. The second ongoing clinical trial it's also recruiting is an intracisterna magna delivery of AAV-HU68. So this is a novel AAV capsid that's very similar to AAV9. With this study, there's no prior conditioning or transplantation, and it's given in the cisterna magna, and the serotype is AAV-HU68. So just very briefly, since I think Krabbe is kind of unique in that we've had this really recent success, I'll just show you a little bit about how we got there from the bench to the bedside. So the preclinical study with the HSCT and the intravenous delivery in the Twitcher mouse was done in David Wenger's group, and he was really able to show that in comparison to bone marrow transplant alone or intravenous gene therapy alone, when he combined the two, there was this synergistic effect. He then continued to do dosing studies to determine the optimal dose and timing between bone marrow transplant and the gene therapy, and was really able to show a very clear dose effect with this 1x dose, and then he increased it fourfold, further resulted in benefit, and then reducing the dose, there was loss of efficacy. We then translated this into the Krabbe dog model where they received preconditioning, the intravenous gene therapy, and we also showed a clear dose response by giving the combination therapy at the 5-fold higher dose. We completely normalized the disease at 2 years of age. And just show you really briefly, because this is some unpublished work, is that compared to an untreated Krabbe dog, when we gave the intravenous gene therapy alone, this was almost 1 year of age, we saw a clear extension in lifespan because the untreated dogs reach endpoint at 16 weeks. However, we did not completely correct the motor phenotype. You can see this dog has weak legs and also has some cognitive dysfunction. In contrast, when we combine the transplant and the intravenous gene therapy at the 5-fold dose, this dog still has an amazing quality of life at 2 years of age, normal nerve conduction velocity, and normal MRI. So this was translated to the clinic, and this, again, is the Forge Biologics study. There's 2 cohorts. One is the low dose of a gene therapy, and then a dose escalation study. And some of this data was just presented last month at the SSIEM meeting. This was the 100-day 80 time point, showing that this was well tolerated, and there's early indications of improvement in brain development and motor function. So the second clinical trial, how did we get to the clinic for that? So the preclinical study for the intracisterna magna of HU68, we used the Twitcher mouse as a proof of concept. We compared neonatal intravenous gene therapy to ICV. So this was a question earlier in the day about, did you compare different routes? To 2 routes were compared. The intravenous did not have as great of effect as going to the ICV, so directly into the cerebrospinal fluid. 3 doses were looked at, and we also did this in a later symptomatic cohort, which I'm not going to show. Again, we translated this to the dog model where we looked at nerve conduction velocity because this is a clinically relevant outcome measure. We also looked at biomarkers of GAL-C and psychosine in the cerebrospinal fluid, and also measured these dogs by MRI, showed that we were able to improve the disease in all of these areas. We then tested this in toxicity and safety in non-human primates, and we were able to demonstrate that this was safe. And this, again, was translated to the clinic this year. So March, the first patient was dosed, and their cohort design is that babies that are between 4 and 9 months of age are enrolled first. Then that same age cohort is going to have an increase of dose. Then younger babies, 1 to 4 months of age, will be dosed with the low cohort, and then those babies will go to the increased dose cohort. So just real briefly, as I'm being told we're out of time, some of the key challenges of Krabbe disease, which I also think are important. Our lowest score on our heat map was the symptomatic patients and current therapies available for them, and also the multimodal therapies. So I relied on some of the other members of the work group to provide this. So thank you to Ernesto Bongarzón. This is 2 substrate reduction compounds have been published in the last 2 years. So in 2020, an ACD inhibitor by Ernesto's group, and in 2021, a CGT inhibitor by Mark Sand's group. They were both able to demonstrate reduction in the brain cytosine levels in the Twitcher mouse. For years, L-cycloserine was used as a SRT for Krabbe, but it's so high up on the ceramide chain that there was a lot of off-target effects, and that was never really able to be translated to the clinic. So could SRT be a therapy that's available for symptomatic patients, or also in combination with gene therapy or hematopoietic stem cell transplantation if cytosine levels are seen to rise in those patients? And then lastly, Kumlash Dev at Duke was interested in showing that they're small molecular weight compounds. So we can take these already FDA-approved compounds, screen them, and he is showing that there's a robust rescue of pathology, but really only a modest lifespan in the mouse model. So how can we combine these therapies with other approved therapies or in symptomatic patients? So our key takeaways very quickly, the successes are really the availability and utility of preclinical models, active ongoing clinical trials, and our key challenges are really trying to find a way to treat the symptomatic patients and to try to get some of these multimodal therapies to the clinic for those therapies that may not be able to comprehensively treat Krabbe. Okay, I tried to get that all into 10 minutes. Sorry, Adeline.
A tour de force. I think we'll have time for just 2 questions. And yeah.
Hello, great summary about Krabbe disease. I have a question. It was mentioned that we are still lacking a natural history of the treated patients. And we are actually still lacking a, how can I say, full understanding of how treatment works. So my question is, as in other lysosomal storage disorders that are treated with transplantation, there are increasing data suggesting that cross-correction is not the main mechanism of action. What would you speculate might be the main mechanism of action?
Thank you for the question. The cross-correction suggested as a main mechanism originally because this is a lysosomal enzyme. But from our observation, our experimental setup, it looks like there are multiple mechanisms present. As you can see from my slide, the uptake efficiency is different. We just providing the calcium, for example, lysosomal enzyme outside, and then just let them take it. It doesn't happen that way. But we got some of the improvement in our preclinical trial. I think that some might be working as a cross-correction. Some others might be directly increase the expression specific cell type because of gene therapy. Also, we have to reduce the inflammation. That is done by the maybe transplantation, because transplantation can provide some of immune cells. So generally, the mutant macrophage microglia cannot process it, but the transplanted cell can properly phagocytize, and the inflammation level could be down. So that's why Dr. Allison and Bradbury presented why that the 2 different therapeutics can increase the lifespan. I think that's my thought. Yeah.
I think we're gonna stop there just because we're already beyond time. Sorry. I encourage everybody to continue the discussion afterwards, but in the meantime, I'd like to invite the multiple sulfatase deficiency group.